Welcome to Chapter 2, Chemical Components of the Cell. In this chapter, we're going to quickly review a lot of chemistry concepts. These are concepts that you should have already covered in your chemistry classes that you took before taking this class. So it's going to be really fast-paced, just a general overview, trying to hit the topics to make sure that you help review or remember, because it's going to play a big role for the rest of the semester in how we're going to talk about these molecules, because chemistry is what drives biology. As much as we want to deny it, we need to make sure we understand it so that we can fully talk about it and understand why the reactions and why the events that we're talking about in the cell happen. So if you need some more help, I've posted some extra videos for you in Blackboard, and please make sure you review them and let me know if you have any questions. So in this quick review, it's going to be all of Chapter 2 at once, and we're going to talk about how chemistry is the backbone of biology. We're going to talk about the 4 families of organic molecules, the importance of water in biological reactions, and the importance of pH. As always, here's our objectives. Please make sure you review these. These are the things that you'll be tested on for the exam, but these are more importantly the big concepts that we need to make sure we're grasping from Chapter 2. And there's this little asterisk here at the bottom about making sure you understand the basic chemical principles of what an atom is and how they work and what electrons are. Make sure that you have those building blocks before you continue on in this class, because it's going to help you tremendously to make sure if you understand those. So chemistry is the backbone of all biology. And to talk about this a little bit, we're going to talk about chemistry in review, the importance of the chemical bond, and types of bonds, okay? So it's really important that you understand some basic concepts of chemistry. What is an atom? The important elements in biology, such as hydrogen, carbon, nitrogen, and oxygen. Those are the really big important elements. But as you can see here on the periodic table, there's a bunch of secondary elements that also play a big role in chemistry. So or in biology. So make sure you understand these elements and how they're playing a role in that. But more importantly, we need to understand how electrons work, how atoms work, how they bond, and then molecular shape and how this plays a role in it. And then lastly, polarity. So make sure that if you don't understand the basic concepts of these, that you go ahead and review them with the supplemental material. As we're going to learn this semester, shape dictates function. Number one thing above everything else, if there's one thing you learn this semester, is that shape of a molecule, shape of a protein, shape of a cell dictates the function of that item that we're talking about. So what determines shape? And they are determined through the bonds of the atoms that make up the molecules. And that's how we get shape. And so when we do that, when we have these shapes, what drives the bond energy, what drives the reactions, what drives interactions. So remember, shape, which is determined by bond, is what controls everything that we know about in biology and that we talk about. So we're going to now talk about a couple of the big bonds that are important in biology. So there's 5 bonds that we need to talk about for biology. They're ionic bonds, covalent bonds, electrostatic attractions, hydrogen bonds, and hydrophobic forces. And we're going to go through those in the next couple slides here so you get a good review real quick on how they are, or how they work and what they are. Ionic bonds, it's all about salt, which is how we make sodium chloride, table salt. And what happens here is that, as you can see from the top image, there's 2 valence shells, one for sodium and one for chlorine. And sodium has 1 electron in its outer valence, and chlorine has 7 in its outer valence. Neither of these atoms are happy because the atoms don't like to have their outside valence empty. And so what will happen here is sodium is going to give its electron to chlorine, which then gives both of them a charge, and now that they're charged, they are attracted to each other. This creates the ionic bond. It's not a super strong bond, but it is enough for an attraction that makes it so these 2 atoms will want to stick together. Conversely, covalent bonds are strong bonds, and this is because the 2 atoms share electrons. These are the strongest bonds we're going to talk about. There are 2 types of covalent bonds that we need to make sure we're aware of. And those are the polar and the nonpolar. In polar covalent bonds, one of the atoms in the molecule is hogging the electrons, and you can see that here with water. In the case of water, oxygen is pulling the electrons towards it, where the hydrogen ions then have this positive charge. And this is what's going to create the attractions for hydrogen bonds that we're going to talk about here in a little bit. Whereas with the molecular oxygen, you can see that they equally share those electrons, so there is no charge across it. And so those are really important to understand, the polar covalent bonds as well as just regular covalent bonds and how that works. Covalent bonds are very strong bonds, and that's the thing that you need to remember, because we don't talk about covalent bonds too much unless we're talking about the building or breaking down of large biological molecules. Most of the interactions that occur in the cell are going to occur through attractions. One of these big attractions is electrostatic attractions. We're going to talk we're going to talk a lot about enzymes and substrates and how they work together, and this is through an electrostatic attraction. Now, the big thing to remember about them, besides the fact that positive attracts to minus and vice versa, its opposites attract, is that the distance between the two molecules matters a lot. If there's a one positive molecule and one negative molecule, but they're very far apart, that attraction is not going to happen. They need to be in close proximity to each other in order to bond. And so this is where Brownian motion and the movement of molecules matters, because it brings these oppositely charged ions into, or molecules into, close proximity to create these attractions so that they can actually come together and act on each other as they need to for whichever type of event that's supposed to happen. Another important attraction is a hydrogen bond. Now, remember back to that water we talked about. When water, or hydrogen in general, generally when it's bonded to an oxygen or a nitrogen or something that's really electrostatic or electronegative, will pull the electrons towards it, which makes that hydrogen positive. That then makes that hydrogen more likely to be attracted to something that's negative. And you can see that here, where the hydrogens are attracted to the oxygens or attracted to the nitrogens, depending on where we're looking. This is how the two sides of DNA come together, is through through hydrogen bonding. So it's really important to understand the hydrogen bonding plays a huge role in how proteins are folded, and we're going to talk about that in Chapter 3. The last one is hydrophobic forces. Basically what happens is there's two molecules and they don't like the water around it, and they come together to create a hydrophobic force. This is what forms the membranes of a cell. The phospholipids have a hydrophobic end and a hydrophilic end, and they come together to stay away from the water, creating the structure that it is, and that's all through hydrophobic forces. So let's talk about the four families of organic molecules. You'll hear them called macromolecules, you'll hear them called large biological molecules. I'm open to whichever one you want to use in this class, but the big thing is we have four main families of organic molecules that we need to talk about. So in this little section of the lecture, we're going to talk about the formation and breakdown of macromolecules, we're going to talk about carbohydrates, we're going to talk about lipids, we're going to talk about proteins, and nucleic acids. So first, let's talk about how macromolecules are formed, or when they're broken down. There's two different, we need to both break them down to harvest the energy from them, and we need to build them up to create structures within the cell. When we want to build a structure, we use a reaction known as condensation, where we remove the water from two monomers to create a dimer. And you can see that happening on the graphic here. When we want to break something down, we will add water to the dimer to break it down into a monomer. And this same thing happens for huge polymers all the way down to dimers. So this process works the same way. So make sure you understand how this works. You should have seen this before in 201. It should have been a big focus of that, so I'm not going to spend too much time here. But make sure you understand condensation reactions, hydrolysis reactions. Make sure you understand which one is which. So carbohydrates, glucose, sugars, they're the primary energy source for the cell. They also serve as a way to modify macromolecules, and they also supply some anchoring support for the cytoskeleton. They don't make the cytoskeleton, but they help the cytoskeleton anchor to different areas within the cell. So that's the big thing about carbohydrates. All of these carbohydrates and this energy within the entire biosphere is coming from solar energy harnessed by plants that is then transferred into carbohydrates or sometimes other macromolecules that are then used by other organisms. So the big thing when you think about carbohydrates is energy source. The next group we're going to talk about are lipids. Lipids provide a variety of functions for the cell. They are a big energy storage molecule, but really, as far as we're going to talk this semester, we need to think about them as forming membranes with the phospholipids, which is the structure you see here on the slide, and cholesterols, which help maintain membrane fluidity, because membrane fluidity is just as important as membrane structure, because if our membranes were to freeze, then our cells would die and not be able to function the way they need to. Hormones and glycolipids are also really big aspects of the lipid family for us as far as cells go, because hormones act as messengers and glycolipids are huge receptors that help our cells bring in a variety of things into the cell to help us function properly. Proteins are probably the most important group as far as number of functions go. It depends on which book you're reading. There's either 8 or 9 functions of proteins. We're going to hit this in chapter 4 where all we talk about is proteins. But for now, I want you to understand the basic 8 groups we have here and understand how they work. All proteins start with amino acids, which have an amino group and a carboxyl group followed by an R chain in the middle. And that's how they're broken down. We have 20 different ones of these. I don't expect you to memorize them, but I want you to understand what an amino acid is and how its functions create the protein we're talking about, especially that R chain. Make sure you understand the importance of R chains. But we're going to get into that in chapter 4 a little bit. So we're putting the cart before the horse a little bit with that. But make sure you understand the roles that proteins are playing in the cell. Lastly, we have our blueprints of the cell. DNA, RNA, our nucleic acids. But we don't ever think about it, but it is a part of it, is that ATP, which is adenosine triphosphate, is also a nucleic acid. It has adenosine in it. And we'll talk about guanine triphosphate a little bit this semester too. ATP and GTP are molecules of energy. So it's important to understand that nucleic acids function not only in just genetic blueprints, but also as a transfer of energy through an activated energy carrier of ATP or GTP. And we'll talk about that a little bit next chapter, in chapter 3, when we talk about the energy needs for the cell. The big thing that we haven't talked about yet is water. Our cells are about 70% water, which means that water is critically important to the cell. And there are 4 important properties of water that we need to make sure we understand. Cohesion, which is how water molecules bond together to form almost a surface. If you've ever put water on a penny and form that bubble, that's surface tension, that's cohesion. It's a huge factor. Another big thing is the ability of water to moderate temperature. If water were not able to take in a lot of energy without raising its temperature, it would be really easy for us to overheat or freeze because of the fact that the water is balancing us out that way. And another big thing is that water is less dense when frozen. If water was more dense, everything would sink to the bottom and it would take forever for us to thaw the water. So it's really important to understand that. And then lastly, water serves as the solvent of life. Most things that we need in life dissolve in water. So it's really important. That's partially why our cells are so much water. So make sure you understand these 4 properties and how they play a role within the cell. And the last thing, and I just want to touch on this briefly because we're not going to go into huge amounts of things, but biological pH. We have a biological pH range of 6.9 to 7.4. And it's important that you understand that range. This is why drugs have to be given within a specific range. It's why everything has to be. Because if we go outside of this, our proteins break down, our cellular processes break down, everything can come to us a halt really quickly. So we need to make sure we stay within this biological range, which is why we have a buffering system within our body as well. But that's why anytime anything is administered to us through IV fluids or anything like that, the pH is critical. So this is the end of our quick review of chapter 2, which is the chemical properties. Please make sure you review those objectives. If you feel like you're missing something that you missed somewhere in your chemistry courses, just let me know and we'll make sure we get it covered. And let me know if you have any questions and I'll see you in class.